Hi everyone. It's my pleasure to present my research paper with titled "The Mouse Testis Tissue Culture Could Resume Spermatogenesis as Same as In Vivo Foundation After Human Spermatogonial Stem Cells Transplantation." This research is a part of my PhD thesis at Talbot Madaris University in Tehran, Iran. For introduction, the spermatogenesis process is a complex process which is started with mitosis of spermatogonial stem cell at the base of seminiferous tubule epithelium, and in the continue we will have the meiosis of spermatocytes and some morphological change of spermatids named spermatogenesis, and finally we will have a spermation that release the haploid sperm cells. This spermatogenesis process is affected by several factors, such as interaction between Sertoli cells and germ cells, interaction between the germ cells, and also some growth factors, and in the function of the interstitial cells like Leydig cells and interstitial cells. And any defect of these factors will result to a different level of infertility. Isolation, culture, cryopreservation, and transplantation of spermatogonial stem cells is one of the future treatments for our patients with some gonadotoxic treatments like chemotherapy and irradiation treatments. After isolation, preservation of spermatogenesis stem cell, we will have two options to induction of spermatogenesis by transplantation. The first one is in vivo spermatogenesis, and the second one is in vitro spermatogenesis induction. For the first one, we will have in vivo transplantation of spermatogonial stem cells, that the first transplantation and successful sperm production was done by Brinster and Zimmerman in 1994. And we have lots of successful transplantation and producing the fertilized sperm in the several years with several researchers. But the in vitro transplantation, we have some advantages such as we are able to observe the transplanted cells every day and every week, useful for study the behavior of stem cells in details, and also we are able to circumvent the immunological reject after transplantation. For material and method of this research, we collect the testicular tissue samples from azoospermia patients, Testis sample, who were referred to Gandhi Hospital IVF Center. The age was between 20 and 60 years old. The exclusive criteria was underlying disease such as diabetes, hypertension, and something like that, and drinking alcohol and drugs, and cell-to-cell syndrome. For preparation of azoospermia mouse model, we have used the 40 milligram of busulfan per kg after intraperitoneal injection. After 4 weeks, you can see the result. The picture number 1 and 3 is the experimental group, which is our azoospermia mouse model, and picture number 2 and 4 is the control group without any busulfan injection. And you can see the seminal fusul and epithelium of seminal fusul is completely destroyed. We will have just the remaining of the Sertoli cells. After isolation of spermatogonial stem cell, we cultured that for 2 weeks, and we actually detect the PLZF protein in the colonies derived from the spermatogonial stem cell culture. Picture A showed the positive reaction to PLZF detection. Picture B is the DAPI staining, and C is the merge of these two pictures, and D is the control, the negative control without the primary antibody. For transplantation groups, we had 2 groups, the in vitro groups and in vivo group. The first one, we removed the host testis from the body of host mouse, and we did the in vitro transplantation out of the body of host mouse, and after transplantation, we fragmented the host testis to small pieces and put on the top of the agar gel to put in the organ culture system. And the second group is in vivo transplantation. We did the transplantation in the body of host mouse and keep the house testis in house mouse and just suture the surgery area and keep that for 2 weeks. After 2 weeks, you can see the H&E staining results. Picture A and B is the in vitro transplantation and in vivo transplantation respectively, and the C is the control group without any transplantation. You can see the host, the spermatogonial cell, which is homed on the base of seminiferous tubule in the in vitro and in vivo group, and there is no cells on the base of seminiferous tubule in the control group without any transplantation. For DIR tracing, you can see the positive reaction of subsidized cells on the seminiferous tubule membrane in the in vitro group and in vivo group, and the DAPI staining and merge of these two pictures. And this one is a control group without any transplantation, and there is no positive reaction. And we have done the immunohistochemistry for the our experiments. After 2 weeks, you can see the positive reaction of cells on the seminiferous tubule membrane to PLZF protein in vitro and in vivo group, the DAPI staining and the merge of these cells. And for morphometric studies, you can see the best result for in vivo transplantation group, which had significant difference with the control and in vitro group. The second level, we had the best result for in vitro transplantation group with high number of spermatogenesis stem cells and high percentage of tubules with spermatogonial cells, and significant difference with control group without any transplantation. And the result of real-time PCR showed that we had almost similar expression of PLZF gene in vitro and in vivo transplantation group. On the other hand, we didn't have any expression of PLZF gene in control group, which didn't have any transplantation. Final conclusion, we are able to say that the human spermatogonial stem cells were successfully transplanted to azoospermic mouse model testis in vitro, and the homing of these cells in the testis was supported by tissue culture conditions. And thank you very much for your attention and for your time. And I will be available on the time of Congress. If you have any question, just let me know, and you can send any question to me. My email is mohaqIq@toh.ca. Thank you very much, and have a good day.